Today the topic of our class is antigen receptors and lymphocyte development. As you know, an individual had more than 10 to the power of 7 clone of lymphocyte. Over the years, 3 hypotheses were recommended by scientists for large number of cell clones. Biddle and Tatum proposed 1 gene, 1 polypeptides. Dreyer and Bennett, 1 immunoglobulin chain coded by 2 sets of genes. 1 set coded variable and 1 set coded constant region of immunoglobulin. And finally, Tonegawa, a decade later, presented documented for gene recombination. The pattern of restriction site in genome of committed B cells in comparison with uncommitted cells showed that restriction site in committed B cells get closer to each other. This document supported DNA recombination occur in committed B lymphocytes. Restriction site is a specific site in genome where the restriction enzyme cut the DNA. You see, 6 kb restriction site, restriction site, restriction site, and restriction site. In committed B cells, restriction sites get closer to each other in comparison with uncommitted B cells. 6 kb, 6 kb, and 2.5 kb. In human, there are 3 locus for coding immunoglobulin chain. 1 locus located on chromosome 14, and it's composed of 1250 kb. There are 2 locus for light chain. 1 locus located on chromosome 2 kappa chain, and it's composed of 1820 kb base pair nucleic acid. And lambda chain located on chromosome 22 and composed of 1050 kb. So, 1 locus for heavy chain and 2 locus for light chain. Subtle investigation of the locus on chromosome 40 showed that variable domain of the heavy chain coded by 3 segment DNA V, D, J. V, D, and J coded variable region of the heavy chain of immunoglobulin, and C segment coded constant region of heavy chain, while variable domain of light chain coded by V and J segment, and there aren't D segment in kappa chain and lambda chain. Variable domain of lambda chain coded by V and J segment, and there isn't D segment. So, variable domain of heavy chain coded by 3 segment, while variable domain of light chain coded with 2 segment. As mentioned in the previous class, heavy chain and light chain variable domain composed of 3 CDR, CDR1, CDR2, and CDR3. and CDR3 are very important in antigen binding. In the immunoglobulin heavy chain, all junctional residue between the arranged V and D segment and D and J segment, as well as sequence D and J, make up the CDR3. CDR3 coded by segment D, J, and nucleotide between D and J, V and D, while CDR1 and CDR2 coded by germline V segment. So only V segment participate in coding CDR1 and CDR2, while D and J segment participate in coding CDR3 in heavy chain. In light chain, V and J segment participate in coding CDR3 in light chain. In light chain, V and J and nucleotide between V and J participate in coding CDR3, and CDR1 and CDR2 coded by germline V segment. Only V segment in light chain, only V segment participate in coding CDR1 and CDR2. First recombination occur in heavy chain, and recombination signal sequence is essential for rearrangement. Recombination signal sequence, or RSS, located in 3' or 5' 3' of V, 5' of J, and flanking both sides of D segment. You can see this sequence is a recombination signal sequence. Recombination signal sequence. Recombination signal sequence located on 3' of V, D, and J, 5', 3'. RSS located on 3' of V segment, 5' of J segment, and both sides of D segment. Recombination occur in hyperacetylation region, and methylation also has a negative effect on recombination. Subtle investigation or subtle study showed that RSS consists of highly conserved stretch of 7 nucleotide heptamer located adjacent to the coding sequence, followed by a spacer of exactly 12 or 23 nucleotide, followed by a highly conserved stretch of 9 nucleotide nanomer. You can see RSS heptamer sequence, spacer 23 nucleotide, nanomer, a spacer 12 nucleotide, and heptamer, nanomer, and spacer 23 and 12 nucleotide. This sequence, named RSS, 2 mechanisms recommended for VDJ recombination. One mechanism is deletion, and one mechanism is inversion. We considered 4 stages for VDJ recombination: synopsis, cleavage, processing, and joining. In synopsis, gene segments are in excess of recombination enzyme RAG1 and RAG2. Cleavage, break between RSS and coding segment. Processing, insertion and deletion of nucleotide at the site of break, and finally joining of two broken ends. You can see synapses, cleavage, processing, and finally joining. 4 stages of VDJ recombination. So RAC1 and RAC2, or recombination activating gene 1 and recombination activating gene 2, are lymphoid specific gene. Notice, lymphoid specific gene. So non-lymphoid cells do not express RAC1 and RAC2. So RAC1 and RAC2 are lymphoid-specific genes, form a tetrameric complex, and participate in VDJ recombination. The RAC1 protein recognizes the DNA sequence at the junction between heptamer and coding segment and cleavage, but it is activated only when complex with RAC2 protein. Coding sequence V, coding sequence J, heptamer. RAC1 and RAC2 complex, RAC1, cut DNA between V and heptamer, J and heptamer. This diagram of light chain. The RAC2 protein may help link the RAC1, RAC2 tetramer to other protein, including accessibility factors that bring this protein to specific open receptor gene loci at a specific time and defined stage. Notice, a specific time and defined stage of lymphocyte development. The RAC1 then make a nick between the coding end and the heptamer. The relase 3' OH of the coding end then attack the other strand, forming a covalent hairpin. After cut the DNA, 3' OH attack to other strand and form hairpin, 3' OH. The signal end does not form a hairpin and is generated as a blunt double-strand DNA terminus that undergoes no further processing. RSS, no further processing. This double-strand break results in closed hairpin of one coding segment being held in opposition to the closed hairpin of the other coding end and two blunt signal ends being placed next to each other. RAC1 and RAC2 hold the hairpin end and the blunt end together prior to the modification of the coding ends and the process of ligation. 4 stage for VDJ recombination: synapses, cleavage, processing, and ligation. Do you have any question? Ahmed, do you have? So, Cu17 and Cu18 are DNA end binding protein and bind to the break and recruit the catalytic subunit of DNA-dependent protein kinase, a double-strand DNA repair enzyme. This enzyme is defective in mice carrying the severe SCID mutation, like RAC deficient mice, SCID mice, fail to produce a mature lymphocyte. So, if this protein deficient in cells, B cells cannot recombinate its genes, immunoglobulin genes. DNA-dependent protein kinase phosphorylates and activates an enzyme called Artemis, which is an endonuclease that opens up the hairpin structure at the coding end. In the absence of Artemis, hairpin cannot be opened and T and B cells cannot be generated. Mutation in Artemis result in the absence of T and B cells. Ligation of the process broken end is mediated by DNA ligase. You see? Hairpin structure, V and J segment, this diagram of light chain. In heavy chain, you can see V, D, and J, but in light chain, only V and J. Hairpin structure. After cutting the DNA, hairpin structure formed, and Artemis, after activating, open hairpin structure, and a sticky end were generated. In processing stage, two types of nucleotide were used, P and N nucleotide. P nucleotide and N nucleotide. P nucleotide were inserted by DNA polymerase according to template strand: thymine, adenine, cytosine, guanine, guanine, cytosine, adenine, thymine. So, P nucleotide were inserted on the base of template strand, but N nucleotide is template independent and were inserted by enzymes named TDT or terminal deoxyribonucleotide transferase. Terminal deoxyribonucleotide transferase inserted N nucleotide. In this figure, you can see a diagram of gene rearrangement of light chain V and J segment. For example, V2 joined together with J. Germline DNA, recombined DNA, and finally RNA. In this figure, we can see different stage of immunoglobulin heavy chain gene rearrangement, V, D, and J. In the first step, D joined to J segment, and then V, D, J. And finally, primary RNA transcript, messenger RNA, nascent polypeptide, and finally mature polypeptide. So, two mechanisms are involved in generation of diversity in B cell or BCR. Two mechanisms: combinatorial diversity and junctional diversity. Combinatorial diversity is multiple gene segments may combine randomly. In the V 100, D 23, J 6 segment. Randomly, these segments join to each other. This phenomenon named combinatorial diversity. And finally, junctional diversity. The largest contribution to the diversity of antigen receptor is made by the removal or addition of nucleotide between V and D, D and J, or V and J segment at the time of joining. P and N nucleotide. P and especially N nucleotide are involved in generation of diversity of B lymphocyte or BCR. So two mechanisms are involved: combinatorial diversity and junctional diversity. Allelic exclusion is a very important event in immunoglobulin gene recombination. For example, maternal chromosome, when used for each change, paternal chromosome was excluded. This event guarantees monospecific receptor which for each lymphocyte clone. As you see, heavy chain rearrangement is a first stage in B cell development. If VDG rearrangement is efficient, VDJ recombination on the other chromosome is not occurred. First stage, we can divide B cell development in different stages: pro-B cell, pre-B cell, immature B cells, and finally mature B cells. And so pro-B cells, we can divide pro-B cells to two stages: early pro and late pro. In early pro, DJ rearrangement occurs on both chromosomes, maternal chromosome and paternal chromosome. After VDJ rearrangement on first chromosome, VDJ arrangement on the other chromosome not occurred. This phenomenon named allelic exclusion. After efficient rearrangement of heavy chain, light chain rearrangement is occurred. In the first step, kappa is rearranged. If kappa gene rearrangement is efficient, lambda gene rearrangement is not occur. This phenomenon named isotype exclusion. So, for light change, we can see allelic exclusion and isotypic exclusion, while on heavy change, only allelic exclusion occurs. If kappa gene on the first chromosome is efficient, on the other chromosome it's not occurred. This phenomenon named allelic exclusion. If kappa gene rearrangement is efficient, lambda gene rearrangement is not occurred. This phenomenon named isotype exclusion. After efficient rearrangement of light change, cell enter the next stage named mature B cell. You can see heavy chain, light change, VDJ segment, V and J segment. Rearrangement, VDG, VDJ, VJ. Primary RNA transcript, messenger RNA, nascent polypeptide, and finally mature immunoglobulin. Two heavy chain and two light chain. Mature immunoglobulin. One of the most important stage in B cell development is pre-B cell. Pre-B cell. Pre-B cell. In this stage, B cell express pre-BCR. The pre-BCR are involved in some important event. First, inhibited rearrangement on the heavy chain loci on the other chromosome, allelic exclusion. 2. Stimulate light chain rearrangement, and finally inhibit surrogate light chain expression. Production of kappa light chain inhibit rearrangement of lambda loci. Lambda loci will undergo recombination if kappa light chain cannot be produced. An individual B cell clone can produce only one of the two types of light chain. At the heavy chain locus, production of kappa or lambda chain from one of two parental chromosomes prevent recombination at the other allele, allelic exclusion. So, immature B cell. Immature B cell, in response to antigen, if mature cell recognize self-antigen with high affinity. Immature B cells, if recognize self-molecule as multivalent, cell undergo receptor editing or apoptosis. In the first stage, cells undergo receptor editing. In receptor editing, antigen recognitions lead to the reactivation of RAG genes. Additional light chain, VJ recombination, and production new light chain, mainly kappa chain. In this condition, the cells express different PCR. If receptor editing is not efficient, cell undergoes apoptosis. If immature B cells no self-reaction, if immature B cells has no self-reactions, cells enter the next stage, mature B cell. In the mature B cell, cell express IgD as well as IgM. So, expression of IgD and IgM indicated B cells is mature B cell. If soluble self-molecule linking to receptor, cells converted to anaerobic B cells. And if low affinity non-cross-linking self-molecule, cell converted to mature B cell clonally ignorant cells. The mature B cells can be divided to three types of B cells. First, follicular B cells. Second, marginal B cells. And finally, B1 cells. Marginal B cells and B1 cells involved in production of natural antibody. For example, if blood group of one person is A, he or she produce anti-B blood group. So, different subset of B lymphocyte: follicular B cells, marginal B cells, and B1 cells. Marginal B cells and B1 cells, its variable domain not variable, but follicular B cells has variable domain of follicular B cell is highly variable. In this figure, you can see different aspects of subset of B cells. For example, when first produced B1 cells in fetus, follicular B cell or B2 cells after birth, marginal zone B cells after birth. Response to protein antigen: B1 cells maybe, why? Follicular B cells and marginal zone B cells, yes. Requirement for T cell help, B1 cell has not. Conventional or B2 cells need help of T cells, and marginal zone B cells sometimes needed. Two immunoglobulin IgM and IgD produced using alternative RNA splicing. Expression of IgM and IgD is mediated by alternative RNA splicing, depends to poly A positions. So, antibody diversity. Three mechanisms are involved. Depends on the stage of B cell development. Before production of mature B cells, combinatorial diversity and junctional diversity involved in diversity of B cell, and after production of mature B cells or after challenging of mature B cells with antigen, somatic hypermutation is involved. So, three mechanisms are involved: combinatorial diversity, junctional diversity, and finally somatic hypermutations. These mechanisms are involved in antibody diversity. The class is finished. Ahmed, do you have any question? So, goodbye.